looking at the heart, its anatomy, and learning about how the heart generates and sends electrical signals throughout to control the heartbeat. You're probably tired of hearing this, but again, we're getting to lab material before we get to it in lecture. And this time it's a little bit, we're even a little bit further away because we haven't finished the blood pressure stuff, which is what we're going to do tomorrow. Depending on how long it takes me to get through the blood stuff we have, if there's 15 minutes or whatever left after that, I will start with the talking about the heart, but it's really going to be kind of the same stuff that we're looking at today. Sort of the basic anatomy. And then we'll get into the rest of it next week. You won't have a lab next week. Wednesday of next week has been switched to follow a Monday schedule, so the 25th. So your lab next week is canceled. And that'll actually help kind of get your lab and lecture schedule a little bit closer together. So I'll probably stop saying that we're doing this today and we're going to get to it in the lecture eventually. So, now, what I'd like to do to that's not where I want to go. To start talking about the anatomy of the heart, first I want to talk about the microanatomy, the cellular structure of cardiac muscle tissue. And so I just pulled this picture up off the internet, so I can have something on the screen here, of cardiac muscle tissue. And I want to remind you what you should have seen in AP1 briefly. In my class, I don't really stress it because we really talk about it now, but in the muscle chapter, in that at that point, the book's comparing the different types of muscle tissue. You should have at least seen this passingly. It's not terribly obvious from this picture, but cardiac muscle tissue is striated, which means that it works on pretty much the exact same principles that you learn for skeletal muscle. Myofibrils that are made out of sarcomeres, the thin filaments and the thick filaments, and actin and myosin and troponin and tropomyosin and all of that. It works on the same principle. The myofibrils are not quite as extensive in cardiac tissue as they are in skeletal tissue, but they are there. And part of the reason why it's kind of hard to see the striations in tissue like we have here on the board is because it's not really the entire cell that's filled with these things. Looking at the picture here on the computer screen rather than the projector screen, it's not really that obvious either. What is obvious, though, and more so if you're looking directly at a picture like this, but we can see it a little bit in this, there are lines going across the fibers occasionally here that sort of stand out. They happen to be in the same direction that the striation should be seen. It's just we don't see the striations here. But I picked this particular picture because there are a lot of these that are very obviously stained well on this tissue. These are what are called intercalated discs. And I'm just going to jump over to this picture here, which is out of the book. And they have a little frame showing a micrograph of this, and they're pointing at intercalated discs. There's how the word is spelled. And then they have a cartoon over here suggesting what they are a little bit more. They're basically the edges of a cell. The cell membrane kind of ends in, as this book depicts it, sort of like egg crate looking fashion. And the next cell, which is not drawn into the picture right here, would fit into it, and they'd kind of interlock. And so when we do see a couple cells up against each other, we see this line representing these sort of interdigitated parts of the cell membrane. Those are intercalated discs. The name just comes from viewing them under the microscope, and the word basically means a disc in between two things. That's what intercalated basically means. And it's where two cells come together. They're important to talk about because of what we're going to learn eventually about their function. But it comes down to a way I like to characterize it, that skeletal muscle and cardiac muscle both have to solve a similar problem, but they do it in very different ways, which is the electrical activity, the action potentials that lead to contraction of these cells needs to be propagated all the way up to the entire organ level. So if you just had a little cell in a muscle here and it contracted by itself, that little bit of contraction isn't necessarily going to translate to the entire muscle contracting. What skeletal muscle does is the muscle cells are in fact the entire length of the muscle. They're multinucleated cells, which points to the fact that they used to be a bunch of small individual cells, and they merged together to make the one giant myofiber muscle cell. And it runs from the origin to the insertion. And so when that cell becomes electrically active and depolarization causes opening of voltage-gated calcium channels, and calcium comes out of the sarcoplasmic reticulum, binds to troponin, moves tropomyosin out of the way, and actin and myosin combine, you get contraction. When that happens, it happens along the entire length of the muscle, so the whole muscle contracts. Skeletal muscles are basically linear organs, point A to point B, from the origin to the insertion, so that contraction just has to be amplified along that single line. The heart is very different because the heart's a three-dimensional muscle, if you will. And so there needs to be independent cells. So what we're looking at here, okay, we see a nucleus right here, and that's the nucleus of this cell. And we can see the extent of the cell where the cell membrane kind of outlines it. And then here's another cell, and it has its nucleus. So these are mononucleated cells, meaning that they're anatomically separate cells. But the electrical activity still has to spread across all of the cells at once. And that's what's happening at the intercalated discs. At the intercalated discs, and the reason why they stain darkly under the microscope, there are a bunch of different proteins that help to hold the two cells together. And there are proteins that kind of help to hold the cells together, but more importantly, they're continuous channels across both cell membranes. I have this depiction, which unfortunately won't be captured for the people listening to this lecture separately, but I have two pieces of tarp, and tarp always has around the edges these little metal holes for tying down the tarp. They're called grommets. So let's pretend each piece of tarp here is the cell membrane of one cell. The gap junctions basically are where channels through one membrane line up with channels through the other membrane, and there's one continuous channel all the way from the intercellular space in one cell to the intercellular space in the other cell. So there are channels in the intercalated discs where ions can move from within one cell directly into the inside of another cell. That means that the cells are electrically coupled. Any ion movements in one cell are going to translate directly into the ion movements in another cell. If sodium outside the cell rushes in to depolarize it when it's going through its action potential, then that sodium is going to spread into the next cell. and it will depolarize that cell. More sodium will rush in, and it'll continue it to depolarize across the gap junctions in the next cell. And that'll depolarize and it'll spread into the next cell. So all of the cells end up depolarizing as one. So electrically, they're acting as a single fibrous structure. Anatomically, structurally, they are independent cells so that they can make up their the three-dimensional shape of the heart. But we see them come together at these intercalated discs. And again, under the microscope, you can see those intercalated discs as these dark little stripes going across some of the fibers. And they're dark. They stain because of all of the proteins that are there to hold the cells together. Not just the gap junctions, but other cell adhesion molecules populate that intercalated disc, and they pick up a lot of stain, so they stand out quite a bit. So, whoops. I want to start with that aspect of looking at. I should have scrolled down on this picture. The gap junctions are depicted here. They usually are found in these the parts of the intercalated discs that run along the length of the cell, so they're kind of parallel to the myofibrils. But then where the intercalated discs interdigitate across the cell, the proteins that we see holding them together, the sort of spot connections from one to the other are called desmosomes, which are introduced in the course and in the book back in the epithelial chapter, the tissue chapter, where it's talking about epithelial tissue. Desmosomes are just point-to-point contacts between cells that hold them together. And they haven't named it, but where one myofibril sort of lines up with the next myofibril in the next cell, there's also some adhesion molecules there sort of linking the cells together, so that, again, because they're electrically coupled and they're going to depolarize together, the contractions are going to be coordinated too, so that the whole thing, the whole muscle contracts as one. It can do that in a three-dimensional structure of the heart, as opposed to the very linear construction of skeletal muscles. But it's the same, it's solving the same kind of problem there. So I wanted to do that to introduce cardiac muscle tissue. We're going to get back to what its electrical activity is all about later on. But what I want to talk about next, which we'll need to understand the electrical activity stuff too, is just the basic layout of the heart. I've just put this picture up on the screen for a second just to show you the heart. We'll come back to this. There's a lot of labels on it, so it gets kind of complex. But I actually want to introduce to you 12 important terms. There are 4 chambers of the heart. Here we go. There are 4 chambers of the heart, which I'll list there in a second. There are 4 major blood vessels that we want to think about, which I'll list in a second. And there are 4 valves. So I want to give you these 12 bits of information, and then we'll look at how they come together in the anatomy of the heart. Now, the 4 chambers, there's right and left chambers, and there are the upper chambers, which are atria, and the lower chambers, which are ventricles. So we have a right atrium, a right ventricle, a left atrium and a left ventricle. Now, let me just switch to the picture real quick. I'll come back to this list if you're writing these words down, but I just want to point these out. So within the heart, there are two chambers in the more posterior positions, which are the atria, that's plural for atrium, and two that are in the lower positions, the inferior positions. Those are the ventricles. There's an atrium and a ventricle on the right side, and there's an atrium and a ventricle on the left side. So those are the four chambers. Those chambers get filled with blood, and then the walls of the chambers are cardiac muscle tissue, which contracts and pumps the blood on to the next chamber. And we'll look at the path that blood takes through all of this. Blood has to enter and then eventually leave the heart, and it does that through some major vessels. There are four of those. Essentially, there's a vessel carrying blood into each atrium, and there's a vessel carrying blood out of each ventricle. That's how they're kind of lined up. There's the vena cava, or vena cava, whatever you want to say. It has two components. There's the superior and inferior vena cava. They attach to the right atrium, and in the list here, I'm putting the vena cava first and the right atrium first in the list of chambers because they correspond. They're bringing blood back from the body to the heart. The superior vena cava brings blood from the upper body, anything above the heart, so head, neck, shoulders, arms. And then the inferior is bringing blood back from anything below the heart. So the rest of the thoracic cavity, the abdominal, pelvic cavities, and the legs. All that comes through the inferior vena cava. Blood leaving the right ventricle leaves through what's called the pulmonary trunk, which is the single vessel that then splits into the pulmonary arteries. And I'll show you that in the picture in a little while. What does the word pulmonary refer to? Lungs. So this is where blood is leaving the heart and going to the lungs. And then coming back from the lungs, we have the pulmonary veins, which bring the blood from the lungs back to the heart at the left atrium. And finally leaving the left ventricle, the blood leaves through the aorta, which is an artery. So again, I'll come back to this screen. You'll be able to see these words again if you're in the process of writing down. But the vena cava coming from the lower parts of the body, the inferior vena cava is seen at the bottom of the picture here, and the superior is seen at the top. That brings blood into the right atrium. Blood will then move from the right atrium into the right ventricle, and then the right ventricle pumps that blood out into the pulmonary trunk, which is the big vessel we see coming out here. That then splits into the left and right pulmonary arteries. So the right pulmonary artery kind of disappears behind the aorta and the superior vena cava, but we see a little bit sticking out here on the end, and the left we see going there. And obviously left and right arteries are going to the left and right lungs, respectively. And then the blood comes back from the lungs by the pulmonary veins. We see two over here, and we can actually see where they open up into the left atrium there. And we see two on the right side, which are located posterior to most everything we see here and attached to the left atrium in a posterior position. There are two pulmonary veins on either side, so there's four total. We don't name or number them necessarily. There's just the two on the left and the two on the right. They never come down into a single vein connecting to the heart there. There has to be two. The blood then collects in the left atrium. It'll eventually move down to left ventricle, and the left ventricle then pumps that up into the aorta, which we see arcing over the top. This is the aortic arch because while it's emerging from the superior surface of the heart, it arches over and ends up descending. We don't actually see it in the picture here, but the aorta would continue down and descend through the thoracic and into the abdominal cavity there. Coming off the top of that, these three branches carry the blood up to the head, neck, and shoulders, which we'll talk about in our next lab, which is 2 weeks from now. But those are the major vessels. The vena cava coming into the right atrium, the pulmonary trunk leaving the right ventricle, the pulmonary veins coming into the left atrium, and then the aorta leaving the left ventricle. And back to the list here. I've set up the lists here so that things correspond. Right atrium is the first chamber on the list, and vena cava is the first vessel on the list. And then the right ventricle comes next, and the pulmonary trunk comes next. The left atrium comes next, pulmonary veins come next. The left ventricle comes next, and the aorta comes next. Now, the other thing I want to mention, if you look at this picture here, the vena cava and the pulmonary trunk going to the pulmonary arteries are colored blue, whereas the pulmonary veins and the aorta are both colored red. You're probably used to red and blue corresponding to arteries and veins, but in fact, the color is just about the oxygenation state of the blood. The right side of the heart is pumping deoxygenated blood, the used blood from the body, to the lungs. So the vena cava is blue because it's carrying deoxygenated blood into the right side, and the pulmonary trunk is blue because it's delivering that deoxygenated blood to the lungs. And then the pulmonary veins in the aorta are red because they're both conveying oxygenated blood. So what's coming back from the heart, back from the lungs to the heart through the pulmonary veins is the recently reoxygenated blood, and then that's going to be pumped by the left side of the heart through the aorta out to the body. If you're used to thinking of veins and arteries in terms of their color, it's not a perfect correspondence. Really, veins are any vessels that carry blood to the heart, and arteries are any vessels that carry blood away from the heart. And very simple mnemonic, if you need to remember that, artery and away both start with A. That's generally how people think of it. But we do have a blue-colored artery and red-colored veins here. Notice also that those are specifically the ones that are called the pulmonary vessels. If you are comfortable thinking of arteries as red and veins are blue, that's a safe way to work as long as you remember the pulmonary vessels are backwards. The pulmonary vessels are carrying blood between the heart and the lungs, either to or from. And so their color is going to be different because of the state of the blood relative to that little circulation with the lungs. We are not going to look at any specific vessels in the pulmonary circuit. I do that partly because then we'll just talk about pulmonary arteries and pulmonary veins generally. And you can remember that the pulmonary arteries are colored blue because they're deoxygenated blood, and the pulmonary veins are colored red because they're oxygenated blood. Now, blue is actually not an anatomically accurate color to say veins are. It comes from, if you look at the veins through your skin, they have a kind of darkish purple color, which became thinking of it as blue. But really, it's just as the blood deoxygenates, the iron oxide reaction that gives it its red color sort of lessens. You don't have completely deoxygenated blood. There's still oxygen in the blood, but it's gone from like 95% oxygenated red blood cells down to 75 or something like that. And that much of a change in the oxygenation state is going to change the color from bright red to a darker purple verging on what seems like a dark blue. It just conventionally we use red and blue in anatomy. So those are the 4 chambers and the 4 vessels. The 4 valves are, I'm going to list again corresponding to where we find them, but it's not going to be perfect. It's not like there's a valve for the right atrium and a valve for the right ventricle like that. Valves come in 2 different groups, which I'll get back to this list in a second, but let me just address this. There are atrioventricular valves, and I should say that atrioventricular is abbreviated AV. And I put that before the word valve because a lot of people think AV stands for atrioventricular valve, but really AV stands for atrioventricular. So they are AV valves. And that name tells you that they're between an atrium and a ventricle. There are 2 of those. On the right side, there's the tricuspid valve, and on the left side, there's the mitral valve, which is also sometimes called the bicuspid valve. But mitral is a more common name for it. Now, it might seem like calling the tricuspid valve is trying to point out that that's important, that it has 3 cusps. But in fact, all valves have 3 cusps except for the mitral valve. So that's the one that its name is actually kind of special. It's just tricuspid is the name that that particular valve was always taken on. Then the other type of valve is called a semilunar valve, which I don't tend to abbreviate it too often, but just for consistency, that would be SL for semilunar. And these are between the ventricles and arteries. And they're named for the artery where we find them. So on the right side, we have the pulmonary valve, and on the left side we have the aortic valve. Actually, I don't need this list here. I've kind of put the valves there. That'll do. Now, I just cut out that list and decided I wasn't going to recreate it. The order that I've put these in doesn't quite correspond to the order that I have them up here, but I've been fairly explicit about where these are found. The tricuspid valve is found between the right atrium and the right ventricle. And the mitral valve is found between the left atrium and the left ventricle. The pulmonary valve, I can't quite get it all on the screen together, is between the right ventricle and the pulmonary artery. The aortic valve is between the left ventricle and the aorta. Okay. So going back to the picture here, the valves are all depicted as kind of whitish in color, representing that they're mostly dense connective, dense fibrous connective tissue. So they kind of stand out in the picture here. The AV valves, the tricuspid on the right side and the mitral on the left side, look very different from the other ones because they actually have little tendinous cords, which are called cordi tendini in Latin, which anchor them to the wall of the ventricle. These little projections coming out of the wall of the ventricle are actually attached to those cordi tendini, which we see on the other side here also. Those are called papillary muscles. Papillary just refers to the fact that they're like little bumps on the inside surface, and they attach there. When the valve is closed, the ventricular wall will start contracting. And as it does that, it'll pull on those tendinous cords. I prefer to just use the English word there, which will actually help to keep it closed. And in lecture tomorrow, I'll talk a little bit more about what's going on there. But that's what we see there. The semilunar valves are called semilunar because they have kind of a half-moon shape to them. Actually, one of the models we have has a better depiction of that. But they do not have tendinous cords because the way that they function, they don't need them. And again, when I talk about valves in lecture, did I say tomorrow? I meant next week. I'll explain how they work a little bit more in depth. This is the pulmonary valve between the right ventricle and the pulmonary artery. The aortic valve we see here in a two-dimensional image like this. It's a little bit oddly depicted. It's actually located directly posterior to the pulmonary valve. You got to kind of think about the three-dimensional shape of the heart. I've gotten the heart models out because I'm going to want you to spend a little time looking at the models so you can see their actual three-dimensional shape. And you can see the aortic valve is actually behind the pulmonary valve there, but they're depicted that way. Now, in looking at in looking at the heart models, what I want you to sort of pay attention to is the direction of blood flow. So from the body, blood flows through the vena cava, inferior for lower body, superior for upper body, into the right atrium, and then through the tricuspid valve into the right ventricle. Actually, let me shorten this up a little bit. I'm going to just abbreviate valve with V, and that's probably not going to be quite enough. We'll see. And then the right ventricle through the, where'd it go? Yeah, I need to make this a little bit smaller. Is that too small to read? I just want to try to fit all of this on one line. You'll see why in a second. Through the pulmonary valve to the lungs. Okay. Now, because I'm typing this in, it has to go right to left how I'm typing it in, but when I'm done, it'll kind of all work out. So let me put this in, and when you see it all up there, it'll make sense. From the lungs, oh, I know what I need to do. From the lungs, the blood's going to go through the pulmonary veins, and I've abbreviated valve with a V, and I'm spelling out veins so that you can tell the difference between them. And this is going to move over so it'll line up in a little bit, but through the pulmonary veins into the left atrium. And then from the left atrium through the mitral valve into the left ventricle. And then from the left ventricle through the aortic valve into the aorta, which is then going to go to the body. So the way I've lined this up, I can't quite put body across both lines, but body to vena cava, inferior and superior components, to the right atrium, through the tricuspid valve to the right ventricle, through the pulmonary valve to the lungs. Whoops, sorry, left one out. Pulmonary valve to the pulmonary trunk. Great. Now, the valve, from the ventricle through the valve to the artery. I'm going to abbreviate pulmonary just to make this stuff fit better. There, that did it. Okay, so from the right ventricle through the pulmonary valve into the pulmonary trunk, which carries the blood to the lungs, where it picks up oxygen, and then the oxygenated blood travels back through the pulmonary veins to the left atrium, and then through the mitral valve into the left ventricle, and then through the aortic valve into the aorta, which carries that oxygenated blood back out to the body. Okay. The way I've written this, it's kind of like just a loop, but in actuality, that's not exactly how it's laid out. Let's see, did I put that picture? No, I didn't. Oh, I did. Sorry, I thought I opened this picture up, but we'll go here.
Just a little thingy.
So I'm just going to this picture in the book. This is a pretty nice representation of the relationship of the heart to the rest of the circulation. Blood coming back from the body, upper body or lower body, through the vena cava, superior and inferior components, into the right atrium, right ventricle, through the tricuspid valve, and then pulmonary valve into the pulmonary trunk, which then splits right and left to go to the lungs, and the lungs send it back through the pulmonary veins to the left atrium, and then through the mitral valve to the left ventricle, and then through the aortic valve into the aorta, which then sends the blood out to the rest of the body, upper and lower. So this is a slightly better depiction of things than what I have written here. This text-wise is just laying out a loop. But notice the way that I've written this, the right atrium and the right ventricle going in one direction, and then the left atrium and the left ventricle are going the other direction. Really, the two atria and the two ventricles should be on the same level, side by side, and then obviously right and left should be on top of each other. So the way I've written this in doesn't represent the anatomical structure of the heart. Instead, a picture like what I was just showing you here, come on, there, does show the actual anatomical positioning of the heart and location of everything, and the circulation going with that. Now, what we're talking about here are two aspects of circulation. There's the systemic loop and the pulmonary loop, or systemic circulation and pulmonary circulation. So systemic circulation means use blood from the body back to the heart, which is then going to get it oxygenated through the lungs, and then back out to the body. Systemic just talks about the relationship of the heart to the body. And then the pulmonary circulation is the circulation between the heart and the lungs. So the deoxygenated blood from the body gets sent to the lungs to be oxygenated, comes back to the left side of the heart, which is then going to be pumped out. So we usually think about the systemic and pulmonary circuits or loops or circulation, however you want to describe it, which are pointed out here. So where are we time-wise?
10:53.
So I would like you to take a little bit of time to look at the heart models just to see the three-dimensional shape of everything. There are 4 models here and 3 models in the back. There's probably more than 7 of you here, but some of you can look at them together. Students will often try to kind of shy away from using the big ones. They look old and clunky and kind of troublesome, but they're actually quite good. Because they're so large, we can see a lot of detail. The pieces seem to kind of fall off a little bit because they are getting old, but it's worth it to look at them. They're the only models we have that represent the heart and its major vessels in relationship to other structures. So we see the trachea and bronchi and the esophagus here, which lies between the heart and the descending aorta. And also it has more of the descending aorta than any of the other models. And all of these little things look like kind of octopus suckers. These are just where small arteries are coming off to go to the thoracic cavity. Depicting this stuff. Let's see. The smallest one here just has one piece that comes off, and you can see all of the chambers and valves and stuff inside there. And then and there's only one of this really small one. And then there's 2 of this sort of medium-sized one where you can open up, take the anterior wall off and see both ventricles, and either atrium has its own wall which you can take off. So what I want you to do when you're looking at this is basically to think about the direction that blood flows and pay attention to the three-dimensional structure of the heart. Think about how left and right isn't exactly just left and right. Left is also kind of posterior and right is kind of anterior. So go ahead and look at those, and then after, I don't know, maybe 15 minutes or so, we'll move on to talking about the electrical part of the heart.